Hello there. I'm Dr. Andrew Pantalano, and today I'm going to be discussing with you our first biochemistry experiment, amino acids and paper chromatography. So we're going to be discussing amino acids, which are very critical components in all organisms. So here we have at the top a polypeptide or a protein. This is composed of many different amino acids. Each of these individual amino acids, this one, for example, I will highlight, will have its own unique R group. And that R group will have very unique chemistry. Let's say if we have these individual amino acids, the amine group and the carboxylic acid group, the N-terminus and the C-terminus respectively. And so these end up being rather polar groups. Because they are so polar, this will take a really intense chemical pull to separate them from the other similar groups on the other amino acids. The one difference between them, though, this R group, which is shown here as the side chain, which makes each individual amino acid unique, is going to be what we target in order to separate these amino acids. So the way that we separate these amino acids is using chromatography. Chromatography is just an old process of separating different compounds based on their different polarities. So we have these mixtures, let's say of amino acids in this case, and we plate them or put them on a stationary phase. In this case, we're going to be using paper, basically just spotting them on a piece of paper. If you were to then take a liquid or a gas, if you use gas chromatography, say we're using liquid chromatography, and we're going to be passing them over the surface, these groups are going to get picked up, and as they get picked up, the difference in pull based on polarity is going to slightly separate them into three different pieces. It's going to be pulled a little harder based on the polarity of each of these amino acids. So now, where you had a mixture of three amino acids, you now have these three individual amino acids which have separated. As you allow the pulling to continue, they're going to separate further and further, and we have essentially isolated them. So by isolating these amino acids, we can then understand more about their quantity and other important factors about them that we might want to know for the biochemical experiment that we're doing. This is shown going from left to right, but in reality, the practice of what we're doing in this experiment in TLC starts from the bottom, that is from the bottom of a beaker or a TLC chamber, and moves vertically up through the paper. So this paper is waking up the eluent or the mobile phase. That is this part. Across the paper, which is the stationary phase, and that is pulling these spots and separating them. So each of these spots will reach a particular distance relative to the total distance that the liquid travels. So we call this the solvent front, and you could call this the sample spot or the analyte. The proportion of those two, A over B, is going to be what we use to compare each of these individual amino acids. Because they will all have a different relationship with the polarity of the paper that they're plated on versus the polarity of the eluent, they will all have a unique RF, retention factor, and that will determine their identity. So we'll be able to use that to identify an unknown by comparing knowns to another unknown spot, seeing if their RF is the same, and then identifying that unknown. Label each of your test tubes with the amino acid sample they will contain. Make sure that you get enough clean pipettes for each amino acid to have its own. Add about 1 milliliter of each amino acid to the test tubes. Less is more, don't use too much. If you get any of the solution on yourself, make sure that you get rid of your gloves and you clean the area. Replace anything that needs to be replaced because cross-contamination is a huge problem. Get yourself a clean piece of Whatman TLC paper. Make sure your hands are clean before you start handling it. With a ruler, you're going to want to make a mark 3 centimeters from the bottom and 1 centimeter from the top. Make them as straight as possible. Make a mark for each place you're going to put an amino acid sample, at least an inch or 3 centimeters apart. You want to label each starting point with the corresponding amino acid and sample that you will be applying. Make sure that your labels are clear. You don't want to mix them up, so you might want to have someone do it for you if you can't do it very well. You want to get some capillary tubes and you want to get a new one for each sample. To plate the sample, what you're going to do is you're going to take the pipette and press it gently to the surface of the Whatman TLC paper. Be careful because they will break and they will stab you and they will hurt you. Once you've spotted all of your samples, give them a few minutes to dry. While those spots are drying, get yourself a graduated cylinder and some 30% acetic acid and measure out 20 milliliters into a 2000 milliliter beaker. Your beaker can be larger or smaller, but you just need to get half a centimeter of solution at the bottom. You will also need 20 milliliters of n-Butanol. Both of these are very polar eluents, and the amino acids are also quite polar. We'll now take the TLC paper and punch a hole in each corner. We will use these corners to create a freestanding cylindrical tube using clean thread. We're going to cover that entire TLC chamber with parafilm. Parafilm is great for this, and it will keep the vapor pressure high in the chamber. Gently lower the TLC plate into the chamber, avoiding splashing or sloshing. Now we will seal the TLC chamber as tightly as possible and allow it to sit for 90 minutes. I decided to also try not puncturing the TLC plate and to allow it to run for a slightly shorter time. This actually turned out really nicely, and moving around and adjusting and manipulating the TLC plate usually causes extra contamination, which this helped prevent. We'll see how it turns out. Once the time is up, we pull out the Whatman TLC paper and we lay it out to dry. Try to keep it as flat as possible and not damage it too much. The solvent front, we immediately record. We should do it as soon as possible to avoid losing where the solvent front ends. We've already talked about what the solvent front is. You want it to be as clear as possible for your measurements and calculations later. The second TLC plate was pulled out of the solution and laid flat to dry, similar to the first. Naturally, you should always mark the solvent front immediately because it will evaporate. Don't get lazy. Due to the shorter development time, the solvent front was naturally lower here, a couple more centimeters. You're going to want to tape down the Whatman paper TLCs to apply the ninhydrin evenly. You're going to spray a 0.5% solution over the Whatman paper evenly and make sure that you do not touch the sprayed surface. You can allow the ninhydrin to develop on the Whatman paper by leaving it, or you can heat it in an oven at 100 degrees Celsius for 30 to 40 minutes, or use a blow dryer. When analyzing your data, you're going to want to record the color and measure the distance from the starting point to the middle of each spot along the vertical axis, as well as the distance from the start point to the solvent front. Pause this video and record all of the data on the top left TLC. What do you think might have gone wrong in these other two plates? Think about it. And please, do anything but be a slob. Clean up after yourself.